my work here. As probably most of you already know, in the mammalian plasma membrane, we have thousands of different lipids and proteins with different local and temporal heterogeneity. But the fundamental mechanisms of how, when, and why they interact with each other mostly remain unanswered. So, since the investigation of different lipid phases in the plasma membrane, it is well thought that in the mammalian cell membrane we have a liquid ordered phase which form certain size domains, and these areas are typically enriched in sterols and sphingomyelin. And there is also another phase, the liquid disordered phase, which is rich in unsaturated fatty acids. So if you think about also proteins in this system, you can appreciate that there are certain post-translational modifications of the proteins which may change the behavior of the protein in regards of these phases. So for example, if you think about palmitylation as an action, the protein gets palmitic acid tails in the membrane, and this protein may change its partitioning from the liquid disordered phase to the liquid ordered phase. So if one wants to work with this system, there are certain limitations and problems, of course, with any other research. So first of all, most of these experiments about the lipid phases were done in model systems, such as bilayers and giant unilamellar vesicles, but results in live cell membranes are rather controversial. Another problem is the size of these domains, since these are roughly 10 or 100 nanometer domains in the plasma membrane. So in order to investigate these domains, we have to have very good microscopes and techniques to see them, like super resolution, for example. And also, if we want to label a lipid, then we may run into the issue that the behavior of the lipid is completely different from the original molecule if it has a huge dye on top of it. So in my research project, my aim was to concentrate on the palmitylation of a transmembrane protein and to recruit these proteins with a technique called micropatterning in live cell membranes. And if we have these proteins recruited in the membrane, which probably induce formation of liquid ordered or disordered phase, then we wanted to probe these domains with different lipids and proteins in the membrane. So first of all, I would like to talk about the technique micropatterning with which we recruited these proteins in the live cell membrane. When we do micropatterning, the main aim is to offer a surface to the cells which has patterned antibodies on top of it. So if we offer this surface to the cell and they grow on them, then probably the proteins will be patterned in the cell membrane. Oopsie. So the steps of the micropatterning is the following. First of all, we put a streptavidin drop on top of a PDMS stamp, how we call it. And after drying, we transfer the streptavidin pattern on top of glass substrate. And after passivation of this substrate with fibronectin, we can basically attach any kind of antibodies, biotinylated antibodies, to the streptavidin pattern. And if the cell is growing on these patches of antibodies, we can recruit special transmembrane proteins or proteins in the cell membrane. So if we have a micropattern cell, we can do single molecule diffusion analysis on lipid probes or protein probes. Here you can see again a micropattern cells as a cross section. And now if we look at this cell from the top, here you can see a turf image of a very cell. And I depicted for you here that if you look at this area is exactly these recruited proteins in the cell membrane, what you can see here in the microscopic image. So the first step in my experiment is always to take an image of these cells. Where are the patterns? How good is the micropatterning? And as a second step, I add lipid or protein probes to the system, and they get incorporated in the membrane. And then I want to track the mobility of these molecules in the cell membrane. So here you see the recruited proteins again. This we usually call this domain as an on area. And next to it we have the off area where there is not so many proteins recruited. And then we can check the mobility of the tracer molecule. And the very important information for us is how is the diffusion of these probes at this on area and outside of the pattern, so at the off area. So here I show you what kind of molecules, obstacle molecules and tracer molecules we chose in these experiments. So we chose a transmembrane protein called hemagglutinin, which by default has 4 palmitylation sites. And we also have a depalmitylated version of this protein. So it's a mutant version where there is 0 palmitylation site at the protein. So these were the transmembrane proteins which we used in the cells, and we recruited them in our experiments here. And then when we added the tracer molecules, I chose certain liquid-ordered phase probes, such as cholesterol, sphingomyelin, and the GPI-anchored protein CD59. And also, we had a liquid-disordered phase probe, such as DOPE, which has unsaturated fatty acid chains in the membrane. So after we have all these ideas, we started to think what can actually happen between a tracer and an obstacle molecule, and we came up with certain ideas such as steric hindrance, which is the most apparent one, since if we have a crowding effect in the cell membrane, then these probes can feel a certain steric hindrance by the recruited proteins. Also, there may be specific binding, so maybe there is an interaction between the probe and the obstacle. But it can easily happen that actually this transmembrane protein has annular lipids forming around them. So in order to find out which scenario is valid, my colleague did some simulation to see what results we can expect. And if you look at these graphs, on the y-axis, what we usually plot is a quotient of the diffusion coefficient of on the on area. So the probe is diffusing here inside, where the proteins are recruited, divided by the diffusion constant outside of this area. So basically, this quotient should be 1 if there is no effect, if the probe can freely diffuse in this area, just as it would do outside of this area. And as we go towards higher densities, so when we have the crowding effect, this number should be lower than 1, meaning that the protein is slowing down in this area. What is very important from these simulations, that if we do the same fitting to our data, actually we can calculate the size, the diameter of the tracer molecule and the obstacle molecule. So we can get an actual number, how big are these proteins, or how big they appear with this analysis system. So if we look at the results, first I depicted here cholesterol, when I measured the diffusion of cholesterol with 2 different cell lines. The first cell line was with the hemagglutinin with palmitylation sites, and the mutant cell, which didn't have any palmitylation sites. And what you should appreciate here, that we didn't see big difference between the diffusion behavior of cholesterol if we had palmitylated or non-palmitylated proteins. We did exactly the same experiments with a GPI-anchored protein, which should be, again, a liquid-ordered phase probe. And as you can see, it's exactly the same kind of behavior. We have a bit of slowing down effect, but there is no huge difference between the palmitylation deficient cell line and the Y-type cell line. Also, I plotted here some numbers for you. So when we calculated the size of the obstacle molecule plus the tracer, for the palmitylation deficient cell line, we got this number, 9.2 nanometer. And for the Y-type cell line, it was 7.8, so it's quite similar. And if you look at the CD59 protein and the obstacle, this number is mostly the same. So after this experiment, I also wanted to try a liquid disordered phase probe, and for that we used DOPE with the same label on top of it, KK114. And doing the same experiments, you can see that here again we got a bit of slowing down effect, and the number is actually very, very similar what we got with cholesterol. So these numbers are rather similar. Yes, here it was 7.8. Okay. Oops. Yes. So after this, actually, we wanted to try other lipid tracers as well, and we used sphingomyelin with an atto dye on top of it. And with this tracer, actually, we got also a quite low number and the same effect, but it was not that significant as with the other lipid probes. So to put these numbers and results in a table, what we depicted here is the case when you have a palmitoylated protein, AJM-GFP, and a depalmitoylated, and again, you can see that there is no big difference between CD59 and cholesterol. And what is most probable, what happens here, that actually there is a steric hindrance between the probe molecule and the obstacle. Also, we wanted to rule out that there is a specific binding maybe between cholesterol and the hemagglutinin and DOPE. So I really wanted to try two different lipids which have very different partitioning behavior, and as you can see, with the same system we got very similar numbers, so we could somehow rule out that there would be a specific interaction between the recruited protein and the tracer. Talking about sphingomyelin, we are not entirely sure if this is steric hindrance or maybe annular lipid formation binding. So in order to find out that, we are currently working on some more experiments with more tracer molecules with exactly the same kind of dye, and hopefully we will get a clearer picture what's going on here. But what I can say for sure is that palmitylation of AGM-GFP did not have a significant effect on cholesterol and CD59 diffusion. And also, comparing cholesterol and DOPE, they show very similar diffusion behavior, albeit they have different membrane partitioning behavior. Also, in order to find out what's going on with sphingomyelin, we are planning to do more experiments with DOPC and DSPC, which should be more likely to partition into the liquid disordered phase. At the end of my talk, I would like to say thank you to my supervisor and my colleagues who always supported me, and of course to our funding agencies, and for your attention. Thank you very much.
Thank you, Gergő, for this very well-structured talk. And are there questions? Well,
with this micro-patterning method, you have these LO and LD phases like immobile within time. Is it true? Or can they a little bit move?
So during the micro-patterning, you make sure that all these molecules are immobile completely.
Okay, so it makes it easier than if you have, for example, GUVs with phase separation, that in this type of experiment it really doesn't move and you're sure what you're probing.
Yes, exactly. So you can, it has a GFP label, so you can actually see them, and they are immobile.
Okay, thanks.
Well, if there are no further questions, then we have a break, and we will meet in 30 minutes at 11:30 at the same place. Thank you very much.